Assalamu alaikum. Today we are going to start and continue the last lecture on spermatogenesis, male gonads. And we were talking about the follicular stimulating hormone, which is indirectly stimulate the spermatogenesis. FSH and testosterone act synergistically mixed together on Sartori cells to stimulate secretion of a hormone, a protein, androgen binding protein, into the lumen of seminiferous tubules and into the interstitial fluid around the spermatogenic cells. So these proteins bind to the testosterone, which is a hormone, keeping its concentration high. Testosterone stimulates the final step of spermatogenesis in the seminiferous tubules. Once the degree of spermatogenesis required for male reproductive function has been achieved, Sartori cells release inhibin, a protein hormone named for its role in inhibiting FSH secretion by the anterior pituitary. So from the anterior pituitary, there is a secretion of inhibin. When there is a completion of the spermatogenesis, then this process has to be stopped and to control the testosterone as well as ABP. So in that way, the inhibin is released from the anterior lobe of the pituitary gland and it inhibits the follicular stimulating hormone secretion by the anterior pituitary. If spermatogenesis is proceeding too slowly, less inhibin is released. If you need more spermatogenesis, you have to reduce the inhibin release from the anterior lobe of the pituitary, which permits more follicular secretion and an increased rate of spermatogenesis. Means it will produce more male eggs and continue the formation of production of spermatogenesis by secreting FSH, follicular stimulating hormone. Testosterone and dihydrotestosterone both bind to the same androgen receptors which are found within the nuclei of the target cell. The hormone receptors complex regulates genes expression. These hormones on the receptor help for the gene expression, turning some genes on and other off. Means they control the genetic expression by inhibiting and stimulating different genes. Because of these changes, the androgen produce several effects. Now we'll see the prenatal development from the sperms and ovums, how the neonatal or prenatal means before the formation of a fetus. We call it prenatal development. Before birth, testosterone stimulates the male pattern of development of reproductive system. That's and the descendant of the testis means in prenatal stage before the birth, the testes are descended in the scrotum, which is moved downwards. Dihydrotestosterone stimulates development of the external genitalia, the outer genitalia of the male, the penis is produced through the dihydrotestosterone stimulation. Testosterone also is converted in the brain to estrogen. The same testosterone is converted into estrogen, which is a female hormone, feminizing hormone, when, which may play a role in the development of certain region of the brain in male. So estrogen is also required to male also, though it is a female hormone, but required for male for the development of certain brain regions in the male. मैंने मैं तो करता था मैं तो कहा था उसमें आगे हाँ कोई काम नहीं, काम पे चक्कर लगाए आप लोग, खाना वाना खाए, मछली लेना तो मछली बनवाओ। हाँ, आइएगा। So prenatal development before birth, testosterone stimulates the male portion of development of reproductive system, not the female, right? That and the descent of the testis means this testosterone will help to descending the testis from upper portion to downward portion into the pouch. Dihydrotestosterone stimulates development of external genitalia. So the external male genitalia is produced through the dihydrotestosterone. This hormone is very essential. Testosterone also is converted into brain to estrogen. This same testosterone is converted into female hormone, that is the estrogen, which may play a role in the development of certain region of the brain in male. Means it's responsible, this estrogen, which is a female hormone, certain region of the brain in male development. Development of male sexual characteristics at puberty, at the time of puberty means 18 years or 15 years, between 15 and 8 years at puberty, testosterone and dihydrotestosterone bring about development and enlargement of the male sex organs and the development of muscular secondary sexual characteristics in moustaches and axillary hairs, pubic hairs. This is all secondary sexual characteristics. The secondary sexual characteristics are traits that distinguish male and female but do not have a direct role in reproduction. Means these are the secondary characteristics which have no role in the reproduction, but they play a role to differentiate between the male and female, their faces, their voice, their structure, their organs, all this, their hair, sorry. These include muscular and skeletal growth that result in the wide shoulder and narrow hips in male, facial and chest hairs with hereditary limits in male, and more hair on other part of the body, thickening of the skin. Male skin is more thickened than the female. Sebaceous gland, oil glands, secretion, and enlargement of the larynx means voice is very uh, tough and harsh, and consequently deepening of the voice means heavy voice in male and soft voice in female. Development of sexual function, androgen contribute to the male sexual behavior, means the male behavior is dependent on the hormone which is androgen and spermatogenesis. So sex drive, libido, means sex drive is also through the androgen in both males and females. Recall that the adrenal cortex is the main source of androgen in female. So from where this androgen is produced? From the adrenal cortex. Where is the adrenal cortex? Adrenal cortex is the suprarenal gland which is above the kidney. Stimulation of the anabolism. Androgens are anabolic hormones, that is they stimulate protein synthesis, means it is increasing the bulk of the muscles in the body because it's a male hormone, so it helps into the protein synthesis. This effect is obviously in the heavier muscles and bones of the most men as compared to women. A negative feedback system regulates testosterone production. When testosterone concentration in the blood increases to a certain level, it inhibits the release of the gonadotropic hormone, releasing hormone by cells in the hypothalamus. So the controlling hormone is reduced from the hypothalamus. Hypothalamus is below the thalamus. As a result, there is a less gonadotropic releasing hormone in the portal blood that flow from the hypothalamus to the anterior pituitary. Gonadotropic in the anterior pituitary then release less LH luteinizing hormones, so the concentration of luteinizing hormones LH in the systemic blood falls. With Less stimulation by LH, the Leydig cells in the testis secretes less testosterone, and there is a return to homeostasis, means return towards the balance. So if there is an excess function of the male hormones, then it has to be controlled through the pituitary and gonadotrophic releasing hormone. Reproductive system ducts in the male, duct of the testis, pressure generated by the fluid secreted by the Sertoli cells pushes sperms and fluids along the lumen of the seminiferous tubules and then into a series of very short ducts called the straight tubes. The straight tubules lead to a network of ducts in the testis called the ratty testis. This testis has got the tubules through which the sperms prepare and comes up towards the external duct. From the ratty testis, sperms move into the series of coiled efferent ducts into the epididymis, which is above the testis. They empty into a single tube called a ductus epididymis. What is epididymis? The epididymis above or over the testis is a comma-shaped organ about 4 centimeter, 1.5 inches long, and that lies along the posterior border of the testis. The plural is epididymi. Each epididymis consists mostly of the tightly called ductus epididymis. The efferent duct from the testis join the ductus epididymis at the lower, larger superior portion of the epididymis called the head. The body is the narrow mid portion of the epididymis and the tail is the smaller inferior portion. As its distal end, the tail of epididymis continues as a duct. Vas difference we call it. The ductus epididymis would measures about 6 meter 12 feet in length if it were uncoiled. So it is in the shape of the coil and distance is its length is very short. As we have mentioned, it's only 4 centimeter. But if you will open it, uncoil it, it is about 20 feet long or 6 meter. The free surface of the columnar cells contains Sertoli cell cilia, which, dis despite their name, are long branching microvilli, not cilia. They increase the surface area for the reabsorption of generated sperms. Connective tissues around the muscles layer attach the loop of the ductus epididymis and carries blood vessels and nerves. Functionally, the epididymis is the site of sperm's maturation, the process by which sperms acquire motility and the ability to fertilize an ovum. This occurs over a period of about 14 days. The epididymis also helps propel sperms into ductus vas differences during sexual arousal by peristalsis contraction of its smooth muscles. In addition, the epididymis stores sperms which remain viable here for up to 7 months. Any stored sperms that are not ejaculated by that time are eventually reabsorbed. Means the sperms which are not comes out, it will reabsorb in that place. Ductus deferens within the tail of the epididymis, the ductus epididymis become less convoluted and its diameter increase. Beyond this point, the ductus is known as ductus deferens or vas deferens. The ductus deferens, which is about 45 centimeter or 18 inches long, ascend along the posterior border of the epididymis through the spermatic cord and then enter the pelvic cavity. Then it loops over the ureter and passes over the side and down the posterior surface on the urinary bladder. The dilated terminal portion of the difference is the ampulla. The mucosa of the ductus deferens consists of pseudostratified columnar epithelium and lamina propria areolar connective tissue. The muscularis is composed of three layers of the smooth muscles. The inner and outer layer are longitudinal and the middle layer is circular. Functionally, the ductus deferens conveys sperms during sexual arousal from the epididymis towards the urethra by peristaltic contraction of the muscular cord. Like, like the epididymis, the ductus deferens also can store sperms for several months. So these are the places, ductus deferens, where the sperms are stored for many years. Any stored sperms that are not circulated by that time are eventually reabsorbed. So, as you have seen, different part of the male sexual organ has a different function. Like I have discussed with you, the ductus differences. I have discussed with you the आपको पैसे तो नहीं चाहिए epididymis. We have discussed about the development of sexual functions. We have discussed about prenatal development, the development of the male sexual characteristic, and we have started from the FSH, follicular stimulating hormone. So these all play different role. The structure, the structure of the whole testis, and within the testis, the structure of epididymis, mass difference, and these all play a storage role and ejaculation role and other things. And the hormones play for the development of FSH and LH, as well as the pituitary gland also secretes certain hormones, androgen, FSH and LH, and the suprarenal gland also secretes androgen hormone. So overall, this so this is all about this discussion for today, and we will discuss in next lecture other topic.